Yeah, so I'm going to talk to you. I am a wildlife ecologist. I'm a data producer, and I created this data pipeline to move my data through in a way that I can use it at a later time. And after we built the thing, we said, damn, this is great. This should be used by other people. This can be a way to bring together other projects. And we've been working, I'd say, the last year and a half on trying to make this thing suitable to network different camera trapping projects together. So I'm going to explain to you why we built it, how we use it, and how we're now trying to evolve it into something else. Okay, so let's get that going. And I know there's a different background here, so some people know everything there is to know about camera trapping. Some people know everything there is about data. We're going to have to cover all those things in order to get through this talk. And okay, so camera traps. This is what the camera traps look like. Camera traps are really not for scientists. Camera traps are for the general public. There are now more camera traps in the US than there are binoculars. There are an amazing number of these things out there bought by hunters and the hunting community to see where the buck is, see where the turkey is, see where their wildlife is. Camera traps are a relatively new development for researchers. I spend most of my time overseas doing surveys, and I don't know how much time we spend looking at some tracker sign trying to figure out what that picture, what that thing is. And it's relatively easy when it's a well-defined trap, but something like this, I have spent so many hours looking at crap on the ground and 4 people arguing over what that crap belongs to, and it's just the most dominant voice in the group. You get to decide, that's a male civet who was heading west at the time that it dropped this thing, and then you write that down because there's no other way to do this thing, and that is so frustrating. Yes, you can use genetics, but it's not timely enough, and it's too expensive for most of these reserves. Usually you're there looking at this thing, trying to figure out what it is. In this case, it's a red fox dropping. But these camera traps can work 24/7. They can sit out there and they can give you some photographic evidence that usually you don't argue about. This is what the animal is. This was the time it was there. This is the date it was there. With that scat, you have no idea whether that thing was dropped yesterday or dropped 2 years ago in some environments. How these camera traps work, for those of you who don't know, it's a heat motion sensor in that it's a hot body moving in front of that camera. So it has to be hot and it has to be moving. So grass doesn't do it, a stationary object doesn't do it, it has to be something that's moving. And in this case, you'd be detecting this animal up in front. You wouldn't be detecting that animal in the corner because it doesn't hit the rays. They're almost like fingers coming out from that camera sensor. And once the camera sensor detects something, sends a message to the camera unit, it just has an autofocus and then usually keeps shooting pictures as long as something is moving in front of the camera. These cameras really replace, I work for the Smithsonian. The Smithsonian is hall after hall after hall of these kind of cabinets full of these kind of animals. This is amazingly useful when you talk about biodiversity. What did biodiversity used to be in certain locations? And it's because you have the specimens and you have the metadata that goes with it. You have the time, the date, the place, who collected it, all this information you need to make that data relevant. The problem is, as a member of the Smithsonian, I can tell you these are almost impossible to collect anymore. You can do it in California or Virginia, but try to go to Brazil and get a sample out of Brazil, you're going to be an old person before that happens. I have ticks. I've been trying to get out of China for 18 months now. Ticks, who cares about ticks? You can't get them out of the country because it's a biological resource that they're not going to let out. That's not the case with these cameras. I can take camera pictures, I can put them in my pocket and bring them home and nobody cares about it. But you can take a picture like this and you can add the metadata to it, and it's no different than a museum specimen. Well, no different. You get about 80% of what you need. You don't have the genetic material, you don't have the morphological measurements, but you do have this animal was here on this date at this time. You can get behavior if you get a sequence of pictures. You can get a lot of information that you can then bring home with you and do your mammal distribution studies, your occupancy detection things. You can do a lot of science with this that you used to do with these museum specimens that you can't collect anymore. And you can do it over a much larger scale usually. So these camera traps are the biodiversity data of the future for a certain suite of mammals. It doesn't work for all mammals. It doesn't work in all circumstances, but it has a much broader range of utility than a lot of the specimen collecting that we used to do. I originally started working on this stuff in Virginia, but pretty quickly I exported to China, where I was kind of required to do some biodiversity surveys. So starting in 2000, we went to a lot of reserves and did a lot of biodiversity surveys, primarily for giant pandas. So you can take a map of a section of, this is a 5-county region, 6-county region in the giant panda range. We can see where we detected giant pandas using those images so that we don't have to worry about sign misidentification, and say this is where the pandas are and this is where the pandas are not. There's a lot of other species that come along. You just can't set the cameras to take pictures of only pandas. You get everything else out there that's with the pandas. And with that, you can do a relative abundance estimates, you can do occupancy things, you can do a lot of science that goes along with those camera data, even for species that you weren't really looking at in the first place. A lot of that has now gone, in the last few years, we've gone to more corridor mapping, looking at corridors between two reserves. These these boxes are camera arrays that are set up between two national parks that are supposed to connect with pandas, and what animals are detected in that corridor area and what are the covariates that they're detected with. And we can use that stuff to create a species distribution model for the pandas. That's what's in green. We can, there's no corridors between those two parks. It's all messed up by roads and houses. Even though it's on paper, we can tell a computer to force there to be corridors between those two parks, and then look at what policy decisions could actually make a corridor. Should we be replanting bamboo? Should we remove the houses? Should we remove the roads? And this over here on the right-hand side is just testing the resistance surface under these different management policies. I think one more of these things. Oh, this is just doing the same thing, not just for pandas, but this is across the whole range of the Chinling Mountains. This is looking at corridors across that area. That's what the bottom map is. And then we can do that for all the other mammals that we capture in those pictures. So again, it gives you a much broader spectrum of animals you can look at. And lastly, now the statistical stuff has been developed, so you don't only do species distribution models, but you can do co-occurrence models. You can look at the probability of detecting one species based on the detection of another species. In this case, we were looking at the impact of these 4 mammals on giant pandas. The Chinese are very worried about takin. Takin is that large goat-like thing on the left, upper left. That animal eats bamboo, and these reserves are set up to save pandas, not takin, and they don't like these takin because they feel they're competing for bamboo. But if you do the co-occurrence matrices, you can see here there's no difference. That's the probability of occupancy for pandas given the presence or absence of takin. There's no difference. They don't impact these giant pandas at all. Wild boar don't impact giant pandas at all. But what does impact is domestic cattle. You find a much lower probability of occurrence if they allow cattle into those parks, and that is, most of the parks do allow cattle. So they should be a lot more worried about cattle than they should about takin. And a synergy thing was Asiatic black bears, which are probably the most hated species in that region of the world because they do a lot of crop damage. The only place you find Asiatic black bears are where somebody's protecting the pandas. Where there's good panda protection, you find black bears. Where there's no panda protection, there's pretty much no black bears. So that's a positive relationship between the two. So all this is just to say we can do good science with camera data. The data is very useful. It gives you presence and absence data, and with presence and absence data you can do a lot of occupancy modeling that allows you to get pretty far down the line. While I was doing the China stuff, I said if I can train a reserve staff in China with a 6th grade education who doesn't even speak my language to do this camera trapping, I can do a guy in Virginia, a woman in West Virginia, somebody in North Carolina. I can use citizen scientists and train them just as easily to do all the stuff I'm having these reserve staff do. So we tested that out on the Appalachian Trail. We took 1,000 kilometers of the Appalachian Trail. We went to there are trail clubs assigned to each section of trail. Went to each of the trail clubs, said, would you put cameras out for us? If we give you the coordinates and the camera, we'll give you a half-a-day training, and you move the cameras along the trail to these coordinates and feed the data back to us. Every trail club said yes. Every trail club had volunteers. We retained these about 95% retention rate of those volunteers over the course of 2 or 3 years. They loved this project. And they fed us all this data for free, which is great too. In that case, what we had is a website where we put in the metadata, and they make a folder of their images and put it in a Dropbox, and we would download those images. That part of the project was a nightmare. It was impossible to keep track of those thousands of images in those folders with that metadata, connecting it up together and using it in any meaningful way. And I said, if I'm going to do this again, the citizen science part is great. This data organization is crap. We got to improve that part of this study and not the citizen science part. So we got ourselves, so I said the citizen science part, we're really following a bird watcher model. Look at everything that's being done with bird data right now. Any of you who've been to the eBird site, their ability to look at the distribution of birds, the change in bird distribution over time, the impacts of climate change on birds, they're doing that because they have 100,000 volunteers collecting bird data across North America and feeding that into a single site. It's the best example of citizen science data, hands down, we have in this country. Why can't we do that with mammals? I like bird people, but I like mammals better. We could do this with mammals. The one thing bird people had that mammal people didn't have is binoculars. Having a tool that allows you to verify what it is you're hearing really made that whole thing possible. These camera traps are the tool that allows the mammal watcher to record what they saw in a way, if they can attach metadata to it, it becomes just like the eBird model. And we could build statewide efforts, countrywide efforts, where we're feeding all that data into a similar place, and we can look at, right now, we look at no mammals across their entire distribution, with the exception of something like giant pandas. That's it. Everything else, we're always looking at a little piece of their distribution, a little piece of their habitat, because nobody's able to bring together data across the entire range of a species the way we can with birds. So this is just, I think this is all the stuff I've just said. What it needs to be like birds is we need a centralized data management. We need to be able to standardize that data across projects. We can't have one person calling something one thing and somebody calling it something else. We can't have the files formatted differently. Everything has to be standardized. We have to be able to share between projects, and we have to have that data accessible to the public. I'm a big believer in, I'm being paid by the federal government to collect data. That data should be accessible to the public and used by the public more than it's used by me. So we created this thing called eMammal. This is the data flow pipeline. And it starts out here with the researchers or the citizen scientists. They use the camera traps. We have a desktop app where it allows them to sort and tag that data relatively easily. They push send. The data goes up to a cloud site. At the cloud site, it undergoes expert review. So some expert looks at it and says, yes, that's a deer. Yes, that's a squirrel. Yes, that's a bobcat. Yes, the dates look good. Yes, it looks like the camera's set up right. Then they approve it. It goes into the Smithsonian digital repository where it's curated. They make backup copies. They do all that kind of stuff with it. And then we feed it out to the eMammal website where the volunteers can see their own data and they can see everybody else's data. And in addition, the researcher can download their data. But allows the complete feedback of that data, and for the website also allows the researcher to communicate with the citizen scientists. So we have a blog post and a discussion board and training videos, things that help those volunteers along and keep them connected. You guys are data people, which I am not. I'm just showing you some more of the detail of how these images are moved along and how the metadata is moved along with it. It has to be packaged up and unpackaged for each one of these stages, and it ends up inside this Fedora repository here and then back out to the Drupal site which is mammal. We spent several days, several long discussions on this shared metadata structure. And we had to get buy-in from most of the major camera trapping organizations. So WCS and CI are also agreeing with the Smithsonian on this is going to be our metadata structure. So when we do projects, we can share across the projects. We have projects, sub-projects, deployments. Deployment is when you set a camera someplace for an amount of time. Inside the deployment, you have images. The images are grouped into sequences. So if an animal stands in front of the camera for a period of time, all those images get linked into a sequence, and the sequence is composed of images, and there's observations, metadata attached to each stage along the way. So you could have this, the project could be California, and each of the sub-projects could be different reserves, or the project could be an individual reserve, and each of the sub-projects could be different projects inside the reserve. So one of them was looking for bobcats, one of them was setting the things for squirrels on trees, somebody was setting cameras on trails. All those different sub-projects can go into the same database because you're capturing enough of the metadata to be able to just do covariates on what the project was actually about, but they all fit inside the same project structure. Okay. So we built this thing with NSF money, and we used it for a project on the East Coast. We gave them a 3-hour training. We had more than 500 volunteers. We gave them all a camera. We gave them coordinates to go to. It's like geocaching in reverse. They have to go to the coordinates, collect the data, bring us the data, go to the next coordinates, go to the next coordinates on their list. And when they finish their list, we give them a new list of coordinates to go to. Across 6 states, 4 million images and metadata. This was all organized with 2 technicians, dealt with all the volunteers and all the data. This is what the desktop app looks like. This is a deployment. This is Tavis's deployment. He was assigned this deployment. He downloads the data on the computer, and he has 16 sequences. He's now looking at sequence number 2. You see the blue box. So that's sequence 2 shown across the bottom, and he's highlighting this picture on sequence 2. So he can look at it, automatically groups all that data together, attaches a position to it, attaches a date and a time, and then what he does is has a drop-down menu. He clicks on what the species is, so there's no misspelling of species, and then he adds the number for that species. And then when he pushes this upload, it goes from his computer up to the cloud site, where then it can get the expert review. You're able to use this star on the bottom to make a favorite photo. So 6 million images, 5.9 of them are terrible, but there's like 100,000 gems in there. You want to know what the 100,000 gems are so that when someone asks, show me your best bobcat picture, you're not looking through the stack. You're just going to the favorite images and bam, those are your best ones. And then we have this bounding box. The bounding box allows us to, if you have a dark, some of those sequences up there are very dark. Maybe you can't see what the animal is. If you push the bounding box, it just looks for change detection in that sequence and says, this is where the change is. So it's put a box around. There's a squirrel inside that box. A volunteer may not see that squirrel. But that bounding box, if they say there's nothing in the image, we ask them to use the bounding box to have the computer look and see if they can see anything inside the image. Then this goes up to the expert review tool. This is it here. It allows you to track where you are. So this is what a PI would use, or the technician in charge of the volunteers. They have some board up on top that tells them how many deployments they set out, how many have been reviewed so far, how many are still left to review, what deployments have been rejected. They can track all that stuff across the top. Here's their latest favorite images. Down below, they can unfavorite something if they don't think it should be a favorite. Like, why is this a favorite image? You know, who knows? This kid probably can't be a favorite image. We got to get rid of that kid picture, so you can unfavorite those things. And down on the side here, you know, most of my experts are actually graduate students. I say, go do this. Do this for 5 hours each day or something. But I have some real species that are hard to tell about. I can assign an outside person as an expert. They can come in and only look at a single species. So this person could look at the coyotes, the 67 coyote sequences, and just ID those coyote sequences. And then get out of the system, and the graduate student can continue with deer, 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 deer. Then it goes into a repository. I know nothing about this repository except that it organizes all my data. Anybody who goes in there usually gets lost. But you come out, at the end of the day, these are the number of projects we have both in country and out of country that are in eMammal. The out of country stuff we've been doing just demonstration projects, so they're not really a lot of data, but there's a lot of data inside the country. We're about 3 million images in December. The website, the eMammal website, this is what it looks like. So it has a bunch of options for you. You can view photographs, you can explore projects, you can browse the data, you can volunteer for the projects. Each project has its own homepage. You can go to its homepage and find out about that project and volunteer for just that project, or you can send a generic email that probably we will never answer about volunteering for anything in the world. There's a carousel that flips through the favorite photographs. There's the blog post, latest blog post entries. It works to communicate with the volunteers and communicate with the public what data is being collected for them. This is an example of what the browse data page looks like. It just, you have maps from an Esri server that we've zoomed in here on the Washington area, and each one of those numbers represent the number of deployments in that area. Not the number of sequences or pictures, but for example, in DC we have 151 deployments. So those are separate camera settings in DC. And if you zoom in on that image, you can see the individual points eventually. And you can click on an individual point and see what the data is for that individual point. Things that are important to people. If researchers are going to use this, a lot of them want an embargo. They don't want the public seeing their data as soon as they see their data. So you're allowed to embargo your project so that the images come up to be seen, but the metadata going with the images stays secret for a certain period of time until you're done with your master's thesis or your PhD thesis or whatever. Eventually, though, this is a federal site, so all your data has to become public. You can't keep it embargoed forever, with the exception of endangered species. Right now, we're sitting with, if it's an endangered species, you will never know the location of that species. You'll know the date it was captured, the time it was captured, but as the public, you won't know the location. The location is assigned to a central point in each project. That's the way we do it right now. And you can add to that list. So if you say, we have some project that's very sensitive about white-tailed deer. They don't want to let other people know where the bucks are, the big bucks are. So all the bucks get assigned to the headquarters because they've made white-tailed deer an endangered species for their project. You have to sign a data provider agreement if you put data into the system. You have to sign a data user agreement if you take data out. The data user agreement said, hey, you didn't collect this data. Somebody else did. You can find, the PI is listed for the project. If you're going to use this data in a scientific way, you should credit the PI. But we can't force that to happen. But we can say this is what you're aware of this before you download this data. 90% of the download is 6th grade kids, 7th grade kids. They're not using the data for publications. Or the volunteers are downloading the data to see their best picture. That's what most of the use is. But they have to sign that data user agreement also. Just to give you an example, here's the East Coast again. We are East Coast people, so we have a lot of East Coast data. We can zoom in on this area, and what I've done is taken two data sets. I had to do this by hand, but I took a Virginia data set, which has core forests in Virginia. That's what all the green is. And I took 3 different eMammal projects and just did a data search that just blended them together. And what I'm showing you are all the camera trap locations in the DC area. The ones in red are the ones that had bobcats detected at the site. The ones in blue are the ones that had coyotes detected at the site. And the yellow sites are sites that had neither species detected. And you can see that bobcats are pretty much confined to some of the bigger forest blocks and don't get very far into the metro area. And coyotes can go all the way into downtown DC. You can find them in Rock Creek Park. I can only do that because I can pull different projects together by having a shared metadata structure and having it sit in a place that it can all be used. That's all the data use, and we built this as individual scientists, and we think other scientists will use it, and citizen scientists find it helpful. But actually, most of my money in the last year and a half has come through education tools. It seems to be most desired by school systems rather than by researchers. Well, we didn't open it up to researchers until this winter. But school systems are very interested in the data as a way to do STEM learning. Some school systems are actually designing their own projects, putting out their cameras. I don't care who submits data to me. If you tell me what your protocols are, I will take your data and put it in this system. You have to have an expert reviewer, just like everybody has to have an expert reviewer, but you can be a 6th grade class and you can contribute data to the Smithsonian website if you follow, if you have protocols that are set and you have an expert review of your data. And that's good. That really allows the schools to get involved. Most of the schools are not collecting their own data. It seems to be almost impossible for a school system to go out in the woods and put up a camera right now. The legal issues are just too much for most school systems. They can put a camera in the schoolyard, but once you get out of the schoolyard, it gets here. But what we've come around to is in Florida, we have money for 25 schools, all to have a volunteer team. It's almost all made up of parents. The parents move the cameras around, and the schools compete against each other on who's getting what pictures based on the parent activity. The schools use the data in their curriculum, so it's data from their county collected by their parents, and they can compare it to other projects in the Smithsonian. This is just an example of what they can do. They can pick a project, they can pick a sub-project, and they have a bunch of data output down at the bottom. I don't think too many researchers would use this, but I think this is primarily a teaching tool for the kids. Essentially, you parse your data, you send it to a sandbox where there's R scripts. The R scripts generate a JSON that comes back with some graph of whatever your parsed data shows. So in this case, we're looking at coyotes. We draw a box around an area. We captured 341 detections of coyotes. We can parse that down further by looking just at a specific time range of all the data that's available. And then in this case, we're graphing the activity of these coyotes versus the activity of bobcats. And it's just to show the students that, in this case, the bobcats, which are the solid line, are much more active during the daytime than the coyotes are. So there are simple analysis things. They can make hypotheses based on looking at the pictures, and then they can test them out with the data. So eMammal is trying to sit in a spot where it's useful for scientists in their individual projects. It's useful as a citizen science tool. It's useful as warehousing and archiving biodiversity data which can never be collected again. You can't go back 10 years and get data that wasn't saved 10 years ago. There has to be some way to put data in a place where people can access it a decade from now or 2 decades from now. And lastly, it can be used by the public in whatever way they want to do it. Do they want to use it as a teaching tool? Do they want to use it for their own little policy decision in their county? You want to see where the bobcats are in your county, you can drill down if you're living in the right area. And you can just be Joe Q from the public, see where the bobcats are and see where the bobcats aren't based on data collections that were done by other people at other times. I'm perfectly fine with that use of the data. They have to undergo the same, caveats that all of us do. Someone will complain about it or not complain about it, depending on how they use it. But the data is for them, and they should be able to see their data and access their data. So I built this thing as a wildlife ecologist who needed some way to organize and use my camera trapping. I am not nearly using the capacity of putting out this data or assembling this data or making use of the potential of this data. And one reason for speaking to a group of you is earlier I spoke to the field station staff from University of California, to start doing camera trapping at all the field stations and use eMammal so they can coordinate across field stations. I also need a center like you to help me with how to use this data, how to display the data, how to use the outreach capacity of this data in a way that will, is beyond my capacity. Let's put it that way. I know this is a good idea. I just have no idea how to implement that good idea. And the rest of this is just telling you the great news. And I don't know if I have to spend a lot of time on this. We have management tools. We can capture the metadata. You can manage thousands of photographs in a project. We have standard data across the projects. We can long-term data store this data, and you can easily get access to this data. The bad news is that right now this thing is costing me money. That data pipeline, it has to move through the Amazon cloud and be put in 3 different buckets as it moves through. Every time it moves from one bucket to the next, and every time somebody packs it or unpacks it, it costs me money. And right now the costs are about $3.20 per deployment month. Deployment month is if you leave a camera out for a month, you get about 400, 500 images. So it's processing those 400, 500 images through the pipeline. So right now I have to charge outside organizations for those Amazon costs. And that's a downer for some. I was talking to Kevin, though, and Michelle, I'm willing to trade Amazon costs for programming skills. That's a trade I'll make. Maybe it's the right audience to do that in. But once you get through the pipeline, the data storage is free, and that dissemination through the website is free. It's the moving it through the pipeline that right now is costing me money. We're trying to work this thing at the local level and at an organizational level. So we've signed agreements with WCI and CI about the metadata structure and the sharing of data. CI is kind of broken off by itself, but the most of us are still together with this is the way we want to display our data. Future things we're working on, all we have the capacity right now is to work on those data downloads, put them in packages that people want to use, like Presence or unmarked R package, so they can do this occupancy analysis. We are going to move this data through Zooniverse or give a project the capacity to move it through Zooniverse. Zooniverse is a crowdsourcing platform where 10 people look at your image and say what it is. So rather than my graduate student going deer, 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 let 10 people over the country look at it and say deer. If they all say deer, it goes in the repository. If 6 say deer, 3 say raccoon, 1 says possum, that's an image I should look at. And that'll save us a lot of time by spending time like that. And lastly, we're probably by the end of this week, well, this is the end of the week, but let's say by the end of next week, eMammal Academy, where we've created now online videos for training all the parts of this system. And we have curriculum from Virginia and Florida. We put that curriculum up there. It'd be great to have California curriculum, but it would be a place where you can put curriculum that's been developed to use eMammal so that school systems can take more advantage of it. So that's the system we have right now. I think it has a lot of capacity. I definitely need help with it, but I think it's the best idea I ever had. And I'm trying to find a way to make it sustainable is the main part. All right, thank you.